and the role of glycans, both the aminoglycans and the glycosaminoglycans in regulation of cancer as well as cancer stem cell growth, and also in the normal stem cell self-renewal. So what we're going to do is try and understand the mechanisms by which at least the glycosaminoglycans, not the aminoglycans, but at least how the glycosaminoglycans regulate some of these phenotypes. So phenotype being the regulation of cancer growth or the stem cell self-renewal. And what we're going to focus on mostly is, its effect on the growth factor signaling, which originates at the cell surface, where most of the glycosaminoglycans are predominantly located or present. So, all right, so I think this could be a challenge. What we want to learn today is understand how heparan sulfate, and of the glycosaminoglycans, I'm mostly going to focus on the sulfated glycosaminoglycans, especially the heparan sulfate, and how they interact with growth factor, ligands, receptors, and their complexes. And the reason for focusing on heparan sulfate is twofold. One, heparan sulfate molecules are quite abundant in the physiological system as well as on the cancer cell. And two, because we saw in the last lecture that heparan sulfate, more so than the chondroitin or dermatan sulfate, played an important role in regulation of cancer stem cell growth. We would also try and understand how this complex and pleiotropic signaling that originates from the interaction of heparan sulfate with various growth factor receptors, participate in regulating a much bigger phenotype, and then understand the structure-activity relationship with respect to the growth factor signaling. Okay, so in this slide, the third slide, you can see I've taken an example of the cancer cells just to illustrate how many various roles that the heparan sulfate molecule plays within a cellular system. And as you may recall, the heparan sulfate molecules are present on the cell surface, but also as soluble molecules within the matrix. And through a variety of actions, you can see that the heparan sulfates can play a role in activating the coagulation cascade, which helps the cancer, which is number A. Number B is, it can regulate the function of the enzyme heparanase, which is involved in cleaving this glycosaminoglycan chain, which can release some of the growth factors and does modulate the growth factor receptor signaling. Or that it can work on some of these endothelial growth factors through regulation of the angiogenic factors and their signaling. And again, it can be involved in immune system, the regulation of immune system cells, their infiltration within the microenvironment as well as their function, and then the classical role of heparan sulfate in regulating the growth factor. Biological functions of the heparan sulfate GAGs that are mediated through its interaction with the ECM molecules. So the heparan sulfate acts at the cell extracellular matrix interface to modulate the cell signaling. And this is possible because of the heparan sulfate molecules, which are expressed by the heparan sulfate chains, which are on the proteoglycan molecules, are extended into the extracellular matrix, where it can interact with a variety of the molecules within the matrix, as well as in a trans fashion with the molecules that are present on the other cells. And these molecules being the growth factors, morphogens, and the cytokines, which play a major role in regulating various intercellular phenotypes with which these heparan sulfate molecules interact. Now, what are the different molecules that HSGAGs does interact with? And this is what's been known in the literature. There are many more molecules which are probably unknown because these molecules through their charge interactions can participate in binding to a variety of proteins and modulate their function. But the most well-studied ones are the growth factors, especially the fibroblast growth factor, the vascular endothelial growth factor, the hepatocyte growth factor, and in the category of cytokines, it could be interleukins, chemokines, the CXC motif ligands or the CC motif ligands, which are very important. It can also interact with the cell interacting molecules. These are the molecules that allow the one cell to interact with the others, such as selectins, and C-selectins and the molecules such as those are important for function of the neutrophils and other immune cells. And then also it can participate in the cell matrix interacting molecules, which are laminin and fibronectin. And these molecules in turn bind to some of the integrin receptors on the cell surface and thus connect to the signaling cascade in the cell. It can also regulate the levels of morphogens, the Wnt and hedgehog, which also play a major role in morphogenesis and in growth and development. And it can also regulate the levels and the function of heparanase enzyme. Moreover, this role of HSGAG has been the most well studied, which is the effect on coagulation system, the various coagulation enzymes and the factors which are targets of heparan sulfate molecules. So how do these heparan sulfate molecules actually regulate the effect of growth factors and the ligand or the receptors? So the heparan sulfate mostly bind to the proteins through electrostatic interactions, and that's shown here. The electrostatic interaction occurs between the negatively charged sulfate groups as well as the carboxyl group in the GAG molecule, which is shown in red, with the positively charged side chain of the basic amino acids. And the most common basic amino acids are lysine and arginine, but sometimes also histidine plays a role in this. So that the lysine and arginine rich areas have been colored in blue. And so that's how this interaction actually takes place. So what are the consequences of this interaction? Once the GAGs, the heparan sulfate GAGs, interact with the protein, it can lead to a variety of different alterations in the protein-protein interactions, as well as the expression of protein or its stability. But the most common consequences of GAG protein interactions are that it promotes the oligomerization of the growth factor, cytokines, and or chemokines. And normally when there is oligomerization, in other words, when these growth factors exist in a monomeric form, they are unable to induce the activation of their receptors. But when you allow the oligomerization, that allows the oligomerization of the ligand as well as the ligand-bound receptors. And in this oligomerized form, most growth factors are able to transactivate each of the monomeric units intracellularly through phosphorylation, but by binding to certain specific adapter molecules, leading to the intracellular signaling cascade. So this is with respect to one growth factor ligand receptor complex, where GAGs can bring them together and sort of allow this formation of oligomerization. The other thing that can happen is that the growth by GAG's ability to bind to more than one type of growth factor ligand or receptor, it can lead to more pleiotropic effect on their activation. And the pleiotropic modulations of the growth factor receptor can activate a very specific type of signaling within the cell, which can regulate a specific phenotype, especially the stem cell or the cancer stem cell phenotype. So this intracellular signaling cascades then can regulate certain key signaling hubs, and these are very specific effects. In other words, certain signaling hubs get activated, certain get inhibited, and that can actually signal a major change in the cell's behavior, leading to regulation of a specific phenotype. The other things that the GAGs could do is potentially stabilize the protein gradient. In other words, in a larger system, certain parts of that tissue can have higher or lower expression of a specific GAG which then binds to a specific morphogen, thus creating a gradient based on either the GAG expression or the ability of the GAG to bind to that particular morphogen. And this gradient are very, very important in organogenesis as well as in the maintenance of normal homeostasis, where, for example, in the intestinal crypt, the stem cells could be at the bottom and then more differentiated cells are at the top. And how do you go from stem cell to a differentiated cell? That all is because of a gradient of certain growth factors, morphogens, which program certain types of cells into differentiation or growth phase. And this can be achieved essentially by differential expression of the GAG molecule, which can then create a gradient for a specific growth factor or morphogen. We'll look at specific examples of this. Or it can present certain cooperative binding molecules to each other and thus regulate, essentially the physiological as well as the pathological functions. And so we'll look at specific examples so you can understand this better. This is one of the examples of where the oligomerization of the growth factor essentially regulates the phenotype. So this is, and the phenotype here is angiogenesis, which is the growth of new blood vessels. This is critical in many different situations, especially cancer growth, because the cancer tissue, in order to support its growth, needs to induce neoangiogenesis, which is the continuous formation of new blood vessels. Wound healing is another example where you need to grow new blood vessels. So in these situations, what happens is the cells which participate in wound healing or cancer growth secrete certain molecules called angiogenic factors, the pro-angiogenic factors like the VEGF, which you see here, which binds to its receptor. Now, when the VEGF monomer is bound to VEGF receptor monomer, usually it does not lead to the activation of signaling. But when they bind to the GAG chain, the ligands can be brought together depending on the spacing of the GAG binding, the protein binding sequences within that particular GAG, that the molecules can be brought together such that it induces this formation of oligomer of VEGF and VEGF receptor. In this case, it's a dimer, but you can imagine that in certain cases it's a multimer. But this leads to the receptors coming together, leads to activation of tyrosine phosphorylation in the C-terminal domain, which is the intracellular domain shown in this box, that they can cross-activate each other. In other words, this acts as an enzyme, the tyrosine, and this is tyrosine phosphorylation of the opposite receptor, and so on and so forth, leading to activation of a signaling cascade. So that's one way it can do that. Now, it's not always that this effect on, because the glycosaminoglycans can bind to more than one receptor type or the ligand type, you can have pleiotropicity, but the pleiotropicity is not always in the same direction. In other words, the GAGs don't just always cause oligomerization leading to activation of receptors and then indiscriminately activate bunch of receptors. In certain cases, and I'm taking an example of the hexasaccharide sequence, which in the last lecture, just to refresh your memory, we showed that in a specific oligosaccharide sequences, especially the hexasaccharide, but not the longer or the shorter chains, actually inhibit the growth of cancer stem cells. So, we examine the effect of this molecule on many different growth factor receptors, and it seems like, it appears that it's not a very non-specific effect. In other words, what it does is activate some receptors while inhibiting the others and causes no effect on some of the other important growth factor receptors. So it's the sum effect of this differential activation sometimes is responsible for modulation of the phenotype. All right, and how can this be utilized? And what is the other consequence? So either through oligomerization or binding to specific growth factor ligand and or receptor, it can either promote signaling or inhibit signaling. And the sum effect of this promotion and inhibition of growth factor receptor activation leads to a specific intracellular signaling, and which can affect, in this case, the cancer stem cell growth. And you can imagine that by creating specific glycosaminoglycan mimetics of a specific sequence and or sulfation pattern, you can create a situation where you either allow binding only to the ligand but not to the receptor, or only one ligand, preventing the oligomerization and thus disrupt the normal physiological or pathological signaling. In this case, because cancer, the signaling would be pathological. Or in a case where the molecules are not coming together, this GAG mimetic, if it's long enough and has appropriate sulfation, can actually bring the receptors together through its own function of oligomerization, promotion of oligomerization, and can lead to specific alteration in this intracellular signaling, which can reverse the phenotype. Okay. So one of the other consequences that I had listed was this maintenance of morphogen gradient. So the previous examples that we looked at was, how the GAGs, through their effect on binding the ligand and the receptors, and either promoting oligomerization or preventing ligand-receptor interaction, can modulate the growth factor signaling. Now, the other function of this is, and these growth factors are usually involved in some growth processes, right? The wound healing, the post-injury healing in different organs, as well as the cancer growth. Now, in the other physiological function where the GAGs do participate or play a major role is the growth and development, as well as the maintenance of normal adult stem cell activity. Now here, you really need to, because the development is a very complex process, which requires coordination between different tissue types in terms of their signaling, such that a specific folding happens, a specific section of the tissue gets transformed into one organ type than the other. Now, to achieve this level of, to actually orchestrate this complex signaling process, you need to generate gradients where some cells are exposed to low levels of morphogen or growth factor, which is shown here in these cells, which are white, and some cells which have very, very high expression, and these cells are shown in red. Now, this type of differential exposure to the morphogen can lead to differential signaling in these cells, and thus each of these cells behave differently. And so you can imagine that you can keep some cells in the self-renewal state and keep them as stem cells, whereas you allow the other cells to differentiate. And how do you exactly achieve that? And as I said, the glycosaminoglycan chains actually play a major role in determining this morphogen gradient. And we'll just look at this example in the next slide and try to understand it in more detail. So as I said, for certain functions such as maintenance of tissue homeostasis as well as the development, you need optimum morphogen gradient. And this optimum morphogen gradient is created by the sulfated glycosaminoglycan molecule. And so you can do experiments where you can actually disrupt one or the other and try and understand the effect on the signaling. And you can see the signaling is shown in this arrow. The bigger the arrow, the thicker the arrow, the more the signaling, essentially. So when you have normal glycan and the normal amount of glycan chain with appropriate sulfation, the yellow dots are sulfation. When you have normal amount of sulfation as well as the glycan chains, they bind to a certain amount of those morphogens and regulate how much of that morphogen is available to the receptor for signaling. And then, when you have a normal physiological condition, you have good signaling. But when you remove the glypican, here it's the proteoglycan, so you attack the proteoglycan, then your glycan chains are no longer available, and these morphogens no longer bind to those glycan chains, are not available for presentation to the receptor, and therefore you have decrease in signaling. So at the same time, you can imagine another scenario where there is too much of a specific type of sulfation that gobbles up all the ligands, so none of the ligand molecules are freely available to bind to the signaling receptor, and then you have less of signaling. Or you could actually cut off this, because the certain proteases can cleave these proteoglycan molecules. Then those proteoglycan molecules have normal levels of GAG change on the surface, and yet because they're not anchored to the cell, that that transfer or presentation of the ligand to the receptor doesn't happen, and that also disrupts the signaling. So you can imagine a variety of ways where the quantity of the glycan chain, as well as the quality, in other words, the sulfation pattern, as well as the molecules that anchor the glycan chains, which are the proteoglycan, interference in any of this or change in the expression of any of this can potentially change the amount of signaling that is mediated by that morphogen. And so you can easily imagine a situation where in a given tissue on different cells, you can have different GAG activity, so to speak, which can be either the quantity of GAG or the quality of GAG, which by itself would be enough to fine-tune this morphogen signaling and lead to very important changes in the phenotype. Okay. So what are these regulators of the HS-GAG and protein interaction? So we believe that HS-GAG and protein interaction, we looked at several examples where either the receptor oligomerization, receptor and or ligand oligomerization, or binding and prevention of ligand to receptor interaction, and or the morphogen gradient creation. This type of GAG protein interactions are important for phenotype. Then now the question is, what are certain factors which may interfere with this process and thus lead to alteration in the phenotype? So one is HS-GAG structure. So, and when we say structure, obviously, structure involves a lot of different aspects. One is just simply the chain length, because these are polymeric molecules. You can imagine a shorter chain might have a different effect than a much longer chain, because the longer chains might have more protein binding domains within that chain. And then it's also the spacing of the different protein binding domains within that GAG chain can be important in this interaction process. So chain length is not just simply the number of disaccharide units, but also how the protein interacting domains are spaced in that chain can actually be one of the most important determinant of the GAG protein interaction. The other more important factor could be the sulfation pattern. And as you know, that the hexosamines within the GAG chains can be sulfated at the N position, or 3 or 6 position, and also the 2 position. The uronic acid can also be sulfated at 2 position. So, and there are specific sulfur transferases that mediate this effect. And so you could have a very unique sulfation pattern on a given glycan chain, and if the cell has more or less of that, then depending on what interactions are favored by a specific sulfation pattern, you could have a differential effect of the interaction of the ligand and the receptor. And besides the sulfation, obviously you have the epimerization, which is either presence of iduronic acid or glycuronic acid can determine the ligand affinity as well. And the spacing I already alluded to. So it's not just the HS-GAG structure, which is the number of disaccharide molecules as well as the sulfation pattern, but also the spacing of the binding site within that polymer, which could be very critical. And then finally, the 3D structure of the GAG chain, which can determine how this GAG protein interaction would be shaped. And eventually, these interactions decide the phenotype. And so this chain length of fine structures can have a much bigger impact on the phenotype. So we're going to look at specific examples of this chain length and fine structural details and how that regulates the GAG protein interaction and perhaps one of the phenotypes. So this is an example. This is a paper in Signaling where they identified that HS GAGs and not any protein ligand actually acts as a sole ligand for the ALK receptors. And the ALK receptors are very important receptors in carcinogenesis, and they are implicated in the growth of many cancers, especially lung cancer, for example. And so in this study, what they showed is that the certain heparin and heparan sulfate molecules were able to bind to the ALK receptor and induce its activation, which can be measured by the amount of phosphorylated form of the ALK receptor. As I showed before, that whenever there is oligomerization of the receptor, the most common thing that happens is intracellular phosphorylation of the C-terminal domain of these growth factors. And so you can actually measure that by specific antibodies that identify those phosphorylated moieties within the receptors, and you can monitor the activation of the receptor this way. And you can see that there is a chain length dependent activation here. It's shown in that line curve, which is the top left panel. And you can see there is a different color for the different oligosaccharide and or polysaccharide chain length. And in this case, this is a traditional GAG protein interaction, wherein the higher the chain length, the better the binding and or activation of the receptor. And in this case, it's the ALK receptor. So you go from the 2 disaccharides all the way to 25 disaccharide units, you see an increase in the activation of the receptor. Now, the sulfation, and this is not looking at specific sulfation pattern, but this is simply looking at removing the sulfate, whether that plays a role. And indeed, if you have a regular heparin molecule, there is good amount of ALK activation. When you have oversulfated heparin, it leads to even better activation. But when you remove the sulfate groups and this desulfated heparin, most of the desulfated heparins that they examined did not show any activation of the receptor. And so was the case for the chondroitin sulfates and the hyaluronan, which are the non-sulfated GAGs. So it appears from this case study is that for certain type of receptor, increasing GAG chain length leads to higher activation of the receptor. All right, I'm going to show you another example. And in this case, it's the 2 receptor systems that we discussed before with the hexasaccharide that showed differential response to the hexasaccharide treatment. In other words, the FGF receptor, we saw an activation, and IGF-1 receptor, we saw an inhibition. So we then asked the same question: if the chain length actually determines the response or the activation of these receptors. And indeed, as you can see, you can appreciate this better on the bar graph, is that the inhibition of IGF-1 receptor, which is the blue bar, is maximum with the hexasaccharide chain. But as you increase the chain length, that effect is lost. And actually, you might achieve the opposite effect, which is the activation of the receptor when the chain length, when we use the unfractionated heparin. And these are soluble GAGs, obviously. But on the other hand, the FGF receptor activation is also highest when there is HSO6, when there is optimum chain length, which is the hexasaccharide in this case. And then after that, with the longer chain, that ability of GAGs to activate FGF receptor goes down too. So in this case, in this particular example, unlike the ALK receptor, here is an example where it's not the increasing chain length which correlates with greater activation or inhibition, but it's the optimum GAG chain length. Because what's not shown in this bar graph is shown here is that the disaccharide, and what's not here in this plot is also the tetrasaccharide. They had essentially not much effect on activation or inhibition of this receptor. So it's not the shorter and it's also not the longer ones, but a specific chain length for the oligosaccharide GAG is critical for the modulation of this growth factor system. So the chain length and the growth factor activation, it's not a simple equation. it is context dependent in the sense that for certain receptors, it is the higher the chain length, the greater the activation, and for others, it is the optimum chain length and sort of a biphasic response to the increasing chain length with respect to the growth factor receptor activation. All right, so that's with the chain length. What about the fine structure? So with respect to the fine structure, you can see that in this cartoon, you have various GAG chains to which specific growth factors bind to. And the black is the FGF growth factor. So the FGF can bind to this red as well as the green chain. And the green chain, because it has optimum spacing for the FGF interacting domain, you can induce oligomerization here, and that leads to the oligomerization of the receptors and hence the signal activation. When you modulate a specific GAG chain, i.e. a sulfation pattern of the GAG chain, then even though some of these chains are able to bind the FGF, it's not able to induce the oligomerization necessary for signal transduction. Hence, what you end up doing is sequestering the ligand without inducing the oligomerization, and hence there is inhibition of signaling. So this creates the fine structures, which would be epimerization, which is the presence of iduronic acid, as well as sulfation, 2-O-sulfation iduronic acid, as well as N, 3, and 6-O-sulfation on the hexosamine can determine the specific type of ligand receptor interaction and hence the signaling process. Okay, so along the same line, looking at specific examples. So the VEGF-A165 is an activated VEGF ligand which requires GAGs to mediate its effect on receptor activation. And it turns out that all important sulfations, the N-sulfated, so the GAG, if it's N-sulfated, 2-O-sulfated, and 6-O-sulfated, you have the best activation of the receptor. And it turns out all of these are essentially equally important for the effect of VEGF, for binding to VEGF and mediating the oligomerization. On the other hand, for hepatocyte growth factor, it can bind to a variety of glycosaminoglycan structure without a clear preference for any of the specific sulfation moieties. And you can see this in this table where KD values are measured for specific GAGs and important ligands. And as you can see in this table, you have heparin molecule, which is desulfated specifically on 2O iduronic acid, as well as 6-O-glucosamine. And looking at the effect on the ligand binding, you can clearly see that FGF receptor, the 2-O-sulfated iduronic acid is essential for FGF2 receptor binding. But in case of HGF, both the 2O and 6O might be playing a role, but a much minor role than the 2O-sulfation plays in FGF2 binding. For VEGF, you can see both 2O and 6O play a major role because removal of either of them significantly affects the binding of VEGF to the heparin molecule. And the BMP6 shows the same profile as HGF. In other words, it has very modest change in the binding after removing specific sulfate groups, and hence it doesn't seem to prefer one type of sulfation pattern. So what you can conclude is that the VEGF ligand requires both the 2O6-sulfated heparin, whereas the FGF2 requires 2O-sulfated heparin in order to have the best binding. All right. And you can do this type of studies for many different ligands and their receptors, and to try and understand what fine structures actually play a role in the binding of the protein the GAGs in the protein. Now, but when it comes to, you can do these studies in vitro. This is essentially an in vitro KD measurement. But when it comes to phenotype determination, it's not as simple. So this cartoon shows that, and you can see that the thiosulfates are depicted in yellow, and the 6-sulfate moieties are depicted in red. And so, no, it's actually, sorry, the red is the 6-sulfate and the yellow is the 2-sulfate. And I just showed you the data that the VEGF molecule here in this previous slide, needs both the 2 and the 6O sulfation in order to have the best binding affinity for the molecule. So what happens is, and this is the conclusion of the experiment, so I'm just showing you the cartoon here. What happens is when you remove this yellow, which is the 2O sulfate, you would expect the effect to be gone, but it's not completely gone, because what happens is when you remove the 2O sulfation by inhibiting the 2O sulfur transferase, for example, you can either knock down the enzyme, or if you have specific inhibitor, you can inhibit the enzyme function. It leads to somewhat compensatory increase in the 6-sulfate moieties, which can partially compensate. So in other words, the protein still interacts, although with lesser affinity, but still maintains some of the function. And so this biological phenotypes cannot be accounted for in the in vitro study, where you would just assume that if you remove one or the other, you're going to see significant decrease in the receptor activation. But this, the feedback system which exists within the cell, where the cell might sense that we're losing critical GAG structures which can affect the activation of the receptor. The signaling has a way of upregulating the other sulfation moiety, which can partly compensate for that effect. All right, so then eventually the modulation of the enzymes that regulate the sulfation pattern, their in vivo effect would be much more complex because of this specific compensatory mechanisms which are at play. And these mechanisms may involve other sulfotransferases as well as the sulfatases, which can eventually try to compensate for the loss of function because of a critical sulfation moiety. Okay. And we examined different ligand families, which was VEGF and FGF. They are not related, but within the FGF family too, there are very specific interactions based on the sulfation pattern. So FGF2, for example, as we saw from the table, prefers 2-sulfated iduronate acid, whereas FGF10 and FGF18 need 6-sulfation, and FGF18 and some of the other FGF would work optimally when both 2 and 6 positions, there are sulfates on 2 and 6 positions. So within the same family too, the binding to different ligands might depend on the fine structure of the GAGs. And the reason why I'm showing you all this example is to illustrate a point that in nature, because of such diversity of the GAG chain, forget about the sulfation pattern, just the number of variations you can have just by having different disaccharide units, variation in the hexosamine or the uronic acid position. You have so many variations, and on top of that, you have this variation with sulfation pattern. You can have quite a bit of diversity in terms of the function mediated by this glycosaminoglycan molecules, and that we should keep in mind. And the additional complexities added by that compensatory phenotype that I showed you is that if you lose one critical sulfation, it could partially be compensated for by the other, especially the molecules that require more than one sulfation for proper binding to the protein. All right. So what are some of the methods that we use to determine the GAG protein binding? So we need to have complementary methods to support and examine the GAG protein interactions, simply because of one of the examples I showed you earlier, is that the in vitro binding affinities might lead you to a different conclusion. But when you apply it in vivo or in cell, the phenotype doesn't necessarily correlate with what you see with the in vitro interactions. So you have to use all these methods because you can't test everything in vivo. You have to make your predictions, then try to validate that in vitro, and then test that in vivo. So in silico methods, the molecular modeling and the molecular dynamics can be used. In vitro, you have you find variety of methods in the literature through which you can determine the GAG protein interactions, quantify the GAG protein interactions. We have commonly used fluorescence in Dr. Desai's lab. Surface plasmon resonance is another method. Nuclear magnetic resonance is also commonly used, and isothermal titration calorimetry is also being used. And you'll find reports of each of these in the literature. And you won't find much evidence for this in the literature, but we are trying to develop methods where we can actually detect GAG protein interactions on the cell surface itself. Because of the complexity involved in GAG protein interactions, you can't study this ideally in the in vitro setting where there is only buffer, whereas on the cell you have the other endogenous GAGs and the other molecules which can compete for these GAG sequences. Okay, so in silico methods, I'm just showing an example how the modeling can be used to determine how this molecule, first of all, of the most likely GAG binding domains, where would your molecule prefer to bind to? And you can do those initial screening followed by molecular dynamic studies, where you can actually, by calculating the binding free energy for each of the residues within a specific GAG binding domain in that protein, you could determine the amino acid residues which are very critical for binding of the GAG to that particular protein. And based on these studies, you would assume that arginine 138 in this protein is critical for the GAG here, which is HSO6. And how these studies can help is that because there is an expansion in the next-gen sequencing in, say, for example, cancers and some other diseases too. Now you have catalogs of mutation patterns that are commonly that exist in a given disease, and you could model those changes. And if there is an overlap between commonly found mutation and the GAG interacting residues, that gives you a clue that that particular residue could be very important for not only GAG binding, but also could be very important moiety to study, because that particular amino acid could be critical for function, not just GAG binding per se, but actually mediating the very important function or a disease phenotype through its interaction with the GAGs. And you can actually do those sort of studies. In vitro studies here, I'm showing an example of change in fluorescence using this method of in vitro binding using fluorescence, and we use the Nile red as a probe. You can either create your ligands which are modified by fluorophores, or you can have this fluorophores which can go and bind your protein of interest. And then upon addition of the GAGs, you could detect a change in fluorescence and come up with a binding affinity. And this is one of the examples where we have measured the binding affinity for the ligand, the receptor alone, as well as when they're in the complex form. And you can derive valuable information where you can see that the KD for FGF2, KD of HSO6 for FGF2 and FGFR1 is low, but when they're in complex, it's even lower, suggesting that that's probably what the GAG sequences may stabilize the complex formation, and you can assume that that would lead to receptor activation. So you can predict some biological functions based on this. This is another example of an MR study of CXCL12, another chemokine, and its interaction with variety of sulfated and non-sulfated GAGs using nuclear magnetic resonance method. And you can actually see the NMR peaks on different amino acid residues and try and figure out which ones are important for interaction with GAGs. And you have this chemical shift measured in parts per million, and you can determine as well as quantitate GAG protein interaction using NMR methods and the other methods that we had listed. But finally, the most important thing to do is to be able to see if you can actually determine binding in a semi-quantitative fashion on the cell itself, because of all the competing GAG and the protein sequences that I mentioned are present in the environment where your GAGs would go and bind to its target. So we developed this, Dr. Desai's lab developed this hexasaccharide sequence, which is labeled with elexaflor, which you'd be able to detect this green signaling, and this is one cell. We don't have a nucleus stained here, but you can, based on the shape of the cells, you can imagine where the cell boundaries are, and you can see very specific punctate pattern of staining by this hexasaccharide. In other words, hexasaccharide is binding to molecules which are in this microdomain, the membrane microdomain, and likely the target is in one of those membrane microdomains. When we knocked out what we thought was one of the targets, we see significant reduction in that hexasaccharide binding. So you could actually verify the specificity of binding using this method. And so that's the last slide I have. So if you use a complement of this method to study GAG protein interaction, you can actually make some conclusions about the phenotype and then actually be able to test the phenotype for your given structure activity relationship, the chain length dependent structure activity relationship or a fine structure, which is the sulfation pattern based structure activity relationship. All right, I'm sorry we started a little late, so we lost 10 minutes, but I have time for questions if you have any.
Okay, thanks, Dr. Patel. Any questions to him? Okay, Sam, go ahead.
I was wondering if you could just briefly go over, I'm still kind of confused about the protein gradients that heparan sulfate is mediating. I mean, is the gradient actually on the cardioglycan per se, or is it I don't know, I guess I'm still confused about that.
All right, so let me just go over that. Yeah, let me go over that slide. So it should be, I don't know what slide number it is, but you can see this slide, right? And you can see the pointer. So I'm going to point out a few things. These are individual cells, and you can see my pointer, correct?
Can you?
Yes, sir.
Yeah.
Okay. So the gradient is not on any specific proteoglycan, but it is on a given cell. So if you can imagine the cell on the left end of this series of cells is a stem cell, okay? And on the extreme right side are the more differentiated cells. You can see a difference in the color. The color just indicates that there is a signaling change. Now, the signaling change is mediated because there is a gradient. In other words, these cells are exposed to, let's assume that it's a bone morphogenic protein. So these cells are exposed to least amount of BMP, and these cells are exposed to the most amount of BMP. Now, this exposure to BMP is not necessarily because of difference in the BMP level. It could be, but it could also be because of the difference in the GAG chains that bind to this BMP molecule. And you can achieve that through a variety of means, and that's what I was trying to show here. You simply can change the amount of proteoglycan, you can change the sulfation pattern, or you can change the number of glycan chains, and all of that would affect how, so in this case, we're not changing the amount of ligand. You can see the same number of ligand molecules. What we are changing is the proteoglycan levels or the GAG chain or the sulfation And all of that can eventually affect how much of the ligand is the cell is able to see. And that's the gradient. The gradient is not just the amount, but how much of the ligand is the cell perceiving. And that's what can be critically regulated by glycosaminoglycans. Is that clear? Or?
Yeah, thank you.
Okay. Any questions from the web? Okay, doesn't look like. So with that, we conclude our first module, right? So